In the last video, I covered how natural selection works and some of the misconceptions associated with natural selection. Today's video is going to start talking about mutations and then continue talking about the other two key microevolutionary mechanisms, gene flow and genetic drift. So let's start talking about mutations. Mutations are really the ultimate source of new alleles and even new genes. And remember, one of the key requirements for natural selection to be able to work is that there's variation in some phenotypic trait that is based on genetic differences. So natural selection can't work without that variation, and variation comes ultimately from mutations. So mutations are oftentimes just this tiny change that can have some pretty profound consequences in evolutionary biology. So to understand mutations, let's do a quick review of what DNA is all about. So DNA is deoxyribonucleic acid. It is a double-stranded molecule of deoxyribose sugar held together by pairs of nucleic acids, purines and pyrimidines, A's, G's, C's, and T's, adenine, guanine, cytosine, and thymine. Now, when mutations typically occur, at least the first scale of mutations we'll talk about, point mutations in indels, which I'll cover here in more detail in a moment, usually occurs during DNA replication. And remember, when DNA replication occurs, when new DNA is made, a current strand of DNA basically unzips, and one strand of that becomes the template for the formation of the new strand of DNA. So that when synthesis is complete, the quote-unquote new DNA is actually half new and half old. And if everything goes right, when the DNA polymerase is making the new piece of DNA, it is using the pairing rules where A's bond with T's and G's bond with C's, such that if the template says, here I have an A, well then a T is going to come into place. Here we have a C, well then a G is going to come into place. So here we have a G, and you can see the C is coming into place. That's if no mutation occurs. But look what we have right here. So here on the template strand, we have a C, so really it should be bonding with a G, but instead we see the placement of an A in this position. That is a mutation. That is what we call a point mutation right there. All right, well, let's look at the consequence of this. The function of DNA is basically the blueprint for the production of proteins in the body. The information in DNA language is converted into messenger RNA, a different type of nucleic acid, in which there are no T's that are used instead in that case. But we use the same basic pairing rules to read the DNA information to make first a messenger RNA strand, which then is, in this process, is called transcription. And then that transcript, messenger RNA transcript, is translated by other forms of messenger RNA, other types of RNA, ribosomal RNA and transfer RNA, to bring in appropriate amino acids in the correct sequence, depending on the information that's ultimately derived from the DNA. So the information is actually contained in these 3-bit words called codons. So here we have a codon, here we have another codon, and another codon. And if you get GUU, well, what's called the universal genetic code says that, okay, well, if I get that, then the appropriate tRNA is going to bring a valine to link in to the neighboring amino acid, in this case alanine, and then glycine, and cysteine, and serine. But this polypeptide chain, or protein, is actually formed based on the information it receives from the messenger RNA, which ultimately is dependent on the messenger that is received from the DNA. And I mentioned the universal genetic code. This is the universal genetic code. And with a 3-letter word, or a codon, with 4 possible letters, there are 64 possible combinations of that. But there are only 20 amino acids, And so that means there's going to be a lot of repetition in here. Now, there are a few of these that are special. So we have some stop codons here that they don't actually, when you get that codon, it doesn't actually signal for a specific amino acid. It actually stops the process of protein formation. There are 3 of these shown here. And then there's one other specific one, a special one called AUG. That codon is the start codon, and it always codes for the beginning of the formation of a protein strand with the amino acid methionine. The remaining 19 amino acids are not randomly distributed in this table, though. If you look at it carefully, you see a big stack of serines, prolines, leucines, threonines, alanine, a little bit less similarity here, but you do see pairs of these together here and here. And what the key to this pattern is the first 2 letters, so C and U, is really all it needs to make leucine. The third position in this codon really doesn't contain any extra information for the production of an amino acid. As long as you have a C and a U, you're going to get leucine. Over here, if you have a C and a C, you're going to get proline. Now, this has important consequences for the probability that a mutation is going to have some phenotypic effect. If you have a mutation in the third position in the DNA that is going to be associated with the third position of a codon, that may not have any effect on the protein that's produced. All right, well, let's go through the different types of mutations. And we're going to start off kind of at the small scale and increasingly get to broader and broader major changes in the DNA. So the simplest type of mutation is called a point mutation. It's also the most common type of mutation. This is where you get a single point in the DNA sequence is changed, so a single nucleotide. So one nucleotide being replaced by another. So an example here, I have a messenger RNA strand where the second codon is CCU. And that would code for these 4 amino acids. If, however, you replace that C with a U, I'm looking at it from the messenger RNA strand position here. Ultimately, the change would have been a little bit upstream in the DNA. So the DNA is where you get the mutation, but the effect would be to change the codon. So just to make that clear. So it would have changed the codon from CCU to UCU. Well, in this case, that would mean that second amino acid would change from proline to serine. Now, the consequence of that we'll talk about here in a moment. Now, there are two basic classes of point mutations. There are transitions in which you have a change from one purine to another, or between pyrimidines from one pyrimidine to another. So remember, the purines are the A's and the G's. So if you go from an A to a G or a G to an A, that is a transition. Or a T to a C or a C to a T, that's also a transition. And point mutations are the most common form of mutation, and within point mutation, transitions are the most common. Transversions are the opposite. So if you go from one chemical class to the other, that's a transversion. So if you go from a purine to a pyrimidine, or a pyrimidine to a purine. And despite the fact that there are more potential combinations of these changes, these tend to occur less frequently. In one study, there were 1.6 transitions for every transversion, but that's going to vary from case to case. But it is generally the pattern that transitions are more common. And here's just a figure from your book that shows you the possible ways that you can get transitions, and the more common ways, the more types of ways you can get transversions, despite the fact that they're not as common. All right, well, let's talk about the effect of a point mutation on a protein's function. In some cases, a mutation can be what's called a silent or synonymous mutation. This is the case where there's absolutely no effect on the protein that's formed, and so the phenotype is not affected. So this is a good example of what I was talking about, the third positions of codons. You get a mutation in the third position of a codon, oftentimes it's going to give you the same amino acid, which then it's going to give you the same protein and the same phenotype. So that's why they're called silent mutations. They really were a mutation. There was a change in the DNA, but there are no evolutionary consequences of this because it produces the exact same phenotypic trait. If you do get a change, we call that a replacement or a non-synonymous substitution. So you're changing one amino acid for another. So if you go from a CAG to a CGG, you go from producing glutamine to an arginine. Now, the effect of this can vary though. In some cases, it can be very significant. So just one amino acid change out of 146 amino acids in the production of hemoglobin can cause sickle cell anemia, which in most cases is going to be a detrimental trait, and natural selection should weed out individuals with that trait that's going to lower their fitness. We will talk about some examples, though, in which, under very specific circumstances, even that phenotype may actually provide some fitness gains. But that's going to be a story for later. The point here is just that one amino acid change can change the phenotype of producing these nice donut-shaped erythrocytes to these sickle-celled red blood cells that have problems clogging in capillary beds and also just have a much lower potential for carrying oxygen, which is the main function of red blood cells. In other cases, an amino acid change may have absolutely no effect or extremely minor changes to the phenotype. I like to use the example of, let's say that you have a mutation in a gene that's associated with the production of proteins that give you more or less body in your hair. That's not really a crucial thing. It's not really going to affect your fitness. It does change the phenotype somewhat, but if it makes your hair more brittle, that's really not a big deal. Okay, so in effect, it really is a replacement substitution, but it's almost like a silent mutation from the perspective that natural selection isn't going to do anything with it. Now, clearly the most serious point mutation is one in which you get a stop codon replacing one that would normally produce an amino acid. So, for example, if you have UCA that gets you serine, but you make one point mutation, a replacement site mutation where you put an A in that codon instead, where you're going to get a stop codon. Depending on where this occurs, but if it happens in the middle of the formation of the polypeptide or protein, it's going to be non-functional. So these typically are going to be nonsense or knockout mutations. All right. The next type of mutation I want to talk about are called frame shift mutations. This is where you are still just affecting at least the transition is the mutation is just affecting one spot of DNA, but it tends to affect the potential of the protein more thoroughly. So a frame shift mutation is when you have an insertion or a deletion of a nucleotide into a DNA sequence. And what this does is it completely changes the reading frame of the codons, and so it can completely change the protein. So for example, if I had this as my original messenger RNA, and it coded for those 4 amino acids, but I put in an additional nucleic acid, it would change the reading frame from the first one being AUG, which it still is, but the second one, instead of being CCU, now is GCC, and then the next one, instead of being CUC, it's UCU. And so everything downstream from the frame shift mutation is now going to be a different amino acid. So all the downstream information is incorrect from the perspective of the original function of the gene, so this is typically going to result in a nonsense mutation. So, what is the rate of mutation? This is a big area of research because mutations, again, are the raw material on which natural selection can operate. And if you look at a per gene mutation rate, it's pretty low. The production of DNA is fairly good. The polymerases that are in charge of making new DNA strands from the template strand don't make many mistakes. And so on a per gene basis, mutations are fairly rare. But you have to think that in most diploid organisms, there are lots and lots of genes. So even a small probability of a per gene mutation accumulates to the entire genome of having a pretty good chance of having a mutation. So one study estimates that possibly 10% of human gametes have mutations that will actually cause some phenotypic effect, some phenotypic change. And this doesn't even count all of those silent substitutions. So when you think about that, probably most individuals do have some mutation compared to their ancestor, their parents. So they do have a different allele at least one locus. But tracking exactly what the mutation rate is can be difficult. Studies that at first just try to track changes in phenotypes to get an estimate of mutation rates are really going to get underestimates. And this is because, in one part, they're completely missing silent substitutions, right? Because those really are mutations, but they're going to give you the exact same amino acid in the exact same place, and so the phenotype isn't going to change. So a mutation occurred, but you're not going to see it if you're just tracking phenotypic changes. And even if it does make a minor phenotypic change, if it's something on the scale of, my hair example, in which your hair is just slightly more brittle because you had one amino acid changed in one hair protein, the gene for production of hair proteins, you're probably going to miss that too, right? Now, on the opposite end of the spectrum are those mutations that cause such major problems that they completely kill off a gamete from being formed. So or kill off an organism in early developmental stages before you can even record it as an individual. So a mutation occurred, but natural selection is wiping that out before it can even become part of the sample. So another approach is to sequence gametes and to get a more accurate result of when and where you see DNA changes. This has been done in the roundworm Caenorhabditis elegans, C. elegans, and it's estimated that there are 2.1 mutations per genome per generation for the nuclear DNA. Now, remember, there are different types of DNA. The nuclear DNA is what we've been focusing on here, but there's mitochondrial DNA in animals, and then in plants we also have chloroplast DNA. And in animals, the mutation rate in the mitochondria can be 10 times that of the nuclear DNA. And in large part, this is due to differences in the repair mechanisms, and also polymerase efficiencies. So we'll see later that there's variation in. So oftentimes, when the mistake is made by the DNA polymerase in forming the new DNA strand, there are repair mechanisms that come behind it and correct some of the mistakes. But there's differences in their efficiencies in being able to find those mistakes and make those repairs. Okay, so yeah, that's what I just said. So DNA polymerases vary in their replication accuracy, and then the repair mechanisms that come along behind that vary in their efficiencies as well. And so that can lead to a variation among and even within species. So, what is a good mutation rate? This may sound like a weird question because if you think about what mutations are doing, most mutations are deleterious if they have a phenotypic effect. I mean, natural selection has molded organisms to be pretty good at doing what they're doing. And if you're introducing random changes into the DNA, random changes in the production of that protein, you're probably going to make something that doesn't work as good in most cases. Now, we talked about silent substitutions, so we can also have neutral mutations happening. But if there is a phenotypic effect, it's typically going to be deleterious. So you may be thinking, well, mutations seem like they're going to be pretty much bad in most cases if they're going to have an effect. And that is true, except the occasional beneficial mutations, those gems in the rough that over the long term of a species may allow a species to adapt to changing environments. The majority of mutations that happen are going to hurt the individual that gets those mutations. If there is a phenotypic effect, it's usually going to be bad. But the rare individual that gets a beneficial mutation, it increases their fitness. That mutant is going to increase in the population. So let's put that into two different environmental contexts. If you are a species living in an environment that is not very stable or is undergoing really rapid, frequent changes, a high mutation rate is actually good for that species. Now, it's not good for most of the individuals of that species, again, because when they get a mutation with a phenotypic effect, it's usually bad. But it provides more opportunities for some of the individuals of that species to get changes in their DNA that may allow them to come up with better adaptations for this changing environment. So it allows for a faster rate of adaptive evolution and may allow that species to persist longer, despite the fact that it really is going to be costly for many individuals of the species. However, if you're a species that is living in a stable environment in which the organisms have had many, many generations of adaptive evolution to fit that environment, natural selection has probably put most of those individuals, pushed them to the point where their adaptations are pretty good fits. And so the population genetics, the more stable it is, the better it is for the individuals, obviously, but it's also the species doesn't need it to persist because the environment is fairly predictable. So if your species in a stable environment, actually a low mutation rate is going to be better. All right, let's change spatial scales here. We were talking about changes in DNA that affect one spot, may change the reading frame, but we're really talking about relatively small scale mutations that would change the alleles associated with a gene locus. Now let's talk about how we can actually add the amount of DNA in larger chunks, producing actually new genes. We'll talk about two ways in which we could get new genes, not just new alleles at a locus, but additional loci. One of those ways is through what's called gene duplication through unequal crossing over. Crossing over occurs in meiosis, where two homologous duplicated chromosomes will line up, and if they do it appropriately, they line up with homologous regions, pairing up such that when they cross over, they're swapping equal amounts of DNA, and they're just replacing one allele for another at all the associated loci. Sometimes gene loci are similar enough where there are pairing irregularities, and this could actually be due to some past errors in DNA replication. So for example, here we have two pink sections that are due to past mutations, and they're fairly closely aligned on this chromosome such that this homologous chromosome and this homologous chromosome don't line up correctly. See, these two pink ones should be over here and these two should be here, but if these pink ones line up here and we see crossing over occur right here, then we're going to have problems. We're going to have, after crossing over, we have this chromatid, which can form a gamete, has extra copies of DNA. It actually has now an extra copy of these three loci. Now, that's at the expense of this one, right? This one actually now has too little DNA, and that probably is going to be filtered out by natural selection because whatever the function, if this is a crucially functioning set of genes, that gamete probably isn't going to make it, or the organism is going to die in early development. But it's usually not going to be a good thing. But let's ask the question, what happens to this individual that is born with this extra DNA? Well, this gamete that has extra regions of DNA is one copy could be free to accumulate mutations. If the other one maintains the original function, this can be thought of as being kind of experimental DNA. And the ultimate result of this could be that the extra copy becomes non-functional. If it accumulates mutations, say a stop codon in the wrong place or any kind of nonsense mutation, it becomes non-functional gene, and we would call this a pseudogene. Its ancestry was from a functional gene, but now it has accumulated mutations that make it no longer functional. So that's what a pseudogene is. Alternatively, it could actually it could become a new gene with a completely new function. If it experiments with different mutations and it accumulates a different polypeptide protein chain that could be used in some other function, it may assume that new function. Or it actually could be used to just produce more copies of the exact same protein that it originally produced, maybe at a faster rate if this is adaptive in some way. And there's actually an example of that. So the AMY1 gene is associated with the production of salivary amylase, which is important for starch digestion. And in human populations that eat a lot of starch, the more copies of this you have, the more efficient you are at extracting this as an energy source. And so there's been selection for keeping more copies of this. So when a mutation occurs in which more copies are produced, that's a fitness advantage to those individuals. In human populations that don't eat as much starch, they tend to have fewer copies of this. There hasn't been such a strong push by natural selection for the preservation of these mutants when they occur. And just to kind of show you where the starting point of this was, that it really was a gene duplication event, probably from just a single gene initially, is in chimpanzee populations. They only have 2 copies of this gene, or in some populations, no functional copies at all. Okay, another form of gene duplication is through the formation of messenger RNA strands that are reverse transcribed back into DNA and then reinserted in the genome. This is called retroposition. And how this is recognized differently from an equal crossing over is these new loci that have been reverse transcribed, when they get reinserted back into the chromosome, they may be in a completely different area from the parent loci from which they were created. In fact, they could even be on completely different chromosomes. Here's an example of this in dog breeding. Some dog breeds that have these extra short legs. This is associated with a gene duplication event due to retroposition and placement of multiple copies of this gene in different parts of the genome. So we just know it happened in this case. And in this case, it actually may have been a good thing that artificial selection by the breeders, they say, "Ooh, that's pretty cool. Let's keep that breed." In nature, this would have been one of those cases where that type of mutation would have been weeded out by natural selection, because it probably wouldn't serve much function in nature. All right, let's go to a completely bigger scale. What happens when organisms can completely duplicate their entire genome? This is called polyploidization, where you add an entire set of chromosomes where you're duplicating. So most organisms you're familiar with are diploid. They have 2 copies of each gene. A polyploid has some multiple of that. So a 4N individual is a tetraploid. An 8N individual is an octoploid. And this is usually a mutation event created by some major error in meiosis, when the gametes should be normally separating from the metaphase plate in telophase and separating the amounts of DNA into the new formed cells. They just stick together and they end up going into one of the gametes that is being formed. This tends to be seen more in plants, and we'll talk about why that is here in a minute. Polyploids usually are instantly genetically isolated, and this can be a mechanism of speciation. And the reason for this is as a say a tetraploid individual, if you then undergo meiosis, the gametes you produce are going to be 2N. So you may still cut them in half, go from 4N to 2N. But now if you have 2N gametes and you mate with other individuals of your species, they're producing haploid gametes. And so you produce a triploid individual, and triploid individuals are usually going to be sterile if they don't have problems just developing initially. Odd numbers of chromosomes just don't work out well for meiosis. So polyploids then have few mating options. Selfing may be the most likely option for species in which this is possible. So in species where they have both male and female reproductive structures, they can just use their own gametes to produce offspring. And this may be why it's so common in plants. Many plants are self-compatible. You might also mate with other polyploids, so offspring that you produce, siblings that are also polyploid, that may be your only option at first. And in some species, polyploid species can persist as all female populations that reproduce through what's called parthenogenesis, where females produce polyploid eggs that just don't need fertilization. So they don't produce eggs through meiosis, they produce eggs through mitosis. And so all of the genes are there that are needed for the individual to develop. And males aren't even needed in those species. Well, what are the consequences of polyploidization from an evolutionary perspective? Well, we have some interesting cases in which it looks like it's actually a good thing. We see a fourfold increase in vertebrate genes in the early ancestor of vertebrates. So if you look at the lineage from non-vertebrate chordates to vertebrates, at that common ancestor, there was a fourfold increase in the number of genes. And if you look at the diversity of vertebrates versus non-vertebrate chordates, like urochordates, there are a lot more species of vertebrates. And so perhaps that extra DNA allowed vertebrates to diversify to the degree that they have. It kind of gave more DNA for morphological exploration and pushing the boundaries and adaptive evolution. To further with that idea, if you look just within vertebrates, the ray-fin fishes, the ancestor of ray-fin fishes, appear to also have experienced an additional polyploid event. And if you compare all of the different major lineages of vertebrates, ray-fin fishes are definitely the biggest group, more species, more morphologically diverse than the other vertebrate groups. And so there does appear to be something to polyploidization and having a genetic capacities for a greater adaptive evolution. Similar data for angiosperms. I mean, if you look what group of plants are the most successful, it's the angiosperms. By far the most group. They represent over 80% of land plants, and many angiosperms come from a polyploid ancestry. All right, well, if you look in different species, if you look at the population genetics of many species, you see that at most loci, the typical population shows most genes are polymorphic, which means at each locus there are multiple alleles existing in the gene pool among individuals of that species. Okay, well, that's clearly the product of mutation. But remember, natural selection is going to be operating on that variability and should be a filtering process, only keeping what's good and getting rid of what's bad. So mutations are creating this genetic diversity, and selection should be reducing it. And I started off this semester talking about how powerful natural selection was. So if natural selection is that powerful of a filter, why do we still maintain all of this polymorphism at the genetic level? Well, I'm posing that question now, and we'll talk about it more in the future. I just want you to be thinking about it now. I'll give you some hints. One hint is The way I've described natural selection has pretty much just been directional selection, and it's been a simplified form. There are other modes of selection that still can be compatible with maintaining genetic diversity. And also, I've already kind of given you a hint of the fact that natural selection isn't always in the same direction within the same species. So when environments change, that changes the filter. And that means that what is adaptive today may not be adaptive tomorrow. And that can also help to preserve polymorphism in a population. But we'll be talking about this more in the coming weeks. I also started this discussion by stating that mutations are random. And I mean that they are not directed by organisms. Organisms can't cause a mutation to occur when they're challenged by changing environment, so that natural selection will have something to operate on. They're completely random with respect to what might be better for those organisms. They don't occur in response to environmental change. They're just mistakes made during the process of making DNA. Now, having said that, yes, certain mutation types are more likely to occur than others. Transitions more common than transversions, and point mutations tend to be seen more than frame shift mutations. But from a functional point of view, with regard to the evolutionary trajectory of a species, mutations are random. Let's now move on to talking about gene flow. Gene flow is actually one of the easier concepts in microevolution to understand, because the name, at least this name, is a very good description of what's going on. Unfortunately, gene flow also has another name. It's also called evolutionary migration, and this can lead to some confusion. So what is gene flow? It's the movement of alleles between populations. And that can be individuals, it could be pollen grains, it could be seeds, it could be spores. Where there might be some confusion is, don't confuse this with seasonal migration that is more of an ecological phenomenon, like seen in some migrating birds. Like right now, warblers are finishing migration through East Texas. Some whales migrate, monarch butterflies, sea turtles. That's not the type of migration we're talking about here. Here we're talking about, that's a seasonal phenomenon. Gene flow is typically a more of a one-time situation where an individual is born in one population and moves permanently to reproduce in another population. Or, again, a seed moves from one population to another. So it's more of a one-time thing. The effect of gene flow is it reduces the difference in allele frequencies between populations, meaning it's a homogenizing force. It reduces the differences between populations of a species. And so we say it reduces the degree of population subdivision. It makes the populations that may be geographically separated more similar to each other from a genetic sense. Within a population, what gene flow can do is increase genetic diversity. And what I mean by that is, remember we just talked about mutations, and mutations are really the ultimate source of genetic diversity, but a mutation that occurs in one population can make it into another population, not just by independent mutation of that allele, but by individuals or seeds or spores from that original population that received the mutation moving to the other one. So it can increase the genetic diversity within a population that receives new alleles from the outside. Okay, so that's basically what I've already described gene flow as being. Let's give an example. If we have, let's say that the green represents some habitat, and the red circles represent different populations. Well, really, clearly, this is just one population. There's lots of overlap between the individual kind of sort of pseudo-populations that really just represents one population. Now here we actually have individual populations that are separated in space. The arrows, however, represent gene flow. There's a high degree of gene flow in this example, compared to this example in which we have the same spacing among individuals in the populations in the habitat, but there's very low gene flow between them. Now this could be differences between the habitat specificity of the species. So if it's a bird species, for example, something like a red-cockaded woodpecker that really is very habitat specific, they're probably not going to fly over long stretches of inhospitable habitat to reach another territory. As opposed to something that's, migratory warbler, yeah, that's no big deal. So the same distance between populations isn't going to be a problem for them. Or, a more extreme example would be like a typical bird going a certain distance between habitat patches versus a snail. They're going to face many more barriers, and it's going to be more difficult for them to travel the same distance. But regardless, in these two examples I have here, one of these effectively from a genetic point of view is like the example on the far left, one basic population from a genetic point of view. Now, is it the one in the middle here or the one on the right? Well, it's the one in the middle, right? Because individuals are moving so frequently among the individual populations within the species in this area that effectively it's just one gene pool. So there's high gene flow that again is reducing the genetic differences among populations. In this example over here, these populations, because they receive so little gene flow, so little genetic connectivity among populations, they're more likely to develop genetic differences between themselves. All right, well, let's look at an example of how gene flow can affect the genetics and the phenotypes associated with different populations within a species. So in Lake Erie, there are different populations of these water snakes. And if you look at the mainland population here in Ohio, you see that, or in Ontario, I guess the data in this case were collected in Ontario, you see that the mainland population, it's mainly banded snakes. So they have these really striking, nice banding patterns, right? So you can really stand out. And on the x-axis here, the A, B, C, and D are basically classifications of the degree of bandedness or unbandedness. So the closer you are to A, you don't have any bands. You get a little bit of bands here, got quite a few more bands here, and then you're heavily banded if you're D. Okay, so that's going to be true for all of these. So if you look at the different populations of these snakes, you see that, again, if you get out on the peninsula but you're still in the mainland, you still have mainly banded snakes, but you're starting to pick up some unbanded snakes. Now you start getting out to these islands and you see a greater mixture of bandedness. When you get out farther away from the mainland, so these that are the farthest from the mainland, that's where you actually have the highest frequency of unbanded snakes. And this actually makes sense from an ecological point of view, because if you study these snakes, you see that on the island, the ones that are unbanded have lower predation rates. So if you have a banding pattern, you're more likely to be picked off by a predator, and so you're going to have lower fitness. Okay, so you may be asking yourself, well, what you just described there is natural selection. Where's gene flow coming into this? Well, the question becomes, why are there so many banded snakes still on the islands? Again, if you have lots of selection, you should be getting rid of these banded snakes. Why are there so many banded snakes? Yes, especially in the more distant islands, you primarily have unbanded snakes, but why are these still being preserved? Well, the answer is we have a balance between opposing forces. The banded snakes regularly are migrating to the island. So this means that there's high gene flow. So gene flow is maintaining banded snakes and the genes for that banding pattern in these island populations, despite the fact that there is a selection pressure to remove them. So this is an example of what we call migration selection balance. Two of the microevolutionary mechanisms that we've discussed are in operation here. And the degree to which migration is more important versus selection is more important will determine the frequency of banded versus unbanded phenotypes and also the associated genotypes on each island. And so that makes sense. The more distant islands had fewer banded snakes. Those that were closer to the mainland preserved more of the banded snakes because they're getting more regular movement of individuals onto those islands, so they have higher gene flow. And here's a figure that kind of mathematically works this out. Don't stress too much about this. Let me just kind of walk you through it a little bit. If we think about a two allele system, to simplify this, where we have A1 representing those with banding patterns and A2 alleles are associated with the unbanded patterns, Q down here on the x-axis is the frequency that you would have A2 at any one time and in one place. Delta Q over here is the change in the unbanded pattern, okay? So if you have strong selection against the banding pattern and little migration, you're going to have, especially at kind of intermediate frequencies of the banding pattern, you're going to have strong selection to get rid of those. Now, clearly, once the unbanded pattern becomes fixed in the population, you're not going to have any change, right? And that's a simplified situation. Now, if you have weak selection pressure, where there's really not that much of a difference between predation rates between the banded and unbanded, and you have lots of migration, well, then you're going to see a great lowering of the unbanded pattern, and you're going to constantly have banded snakes filling up this population, and you're going to see a great decrease in the unbanded snakes representation. Gene flow, it plays a huge role in conservation genetics and how we can think about preserving endangered species. And here's a good example of this. If you look at greater prairie chicken populations in the state of Illinois, historically, so this is a beautiful bird that has some really dramatic breeding behaviors on these traditional breeding grounds called leks. If you look historically, this population, the populations of this species have drastically decreased in the Midwest and the Great Plains. So if you look in the early 1800s, most of the state had populations of prairie chickens, and then there was a severe reduction in the populations due to agricultural modification of lands and separation of species. And so in the mid-90s, you just had these two populations, and they're isolated to a great deal. You're not going to have prairie chickens moving between these two relict populations. And so that is going to greatly reduce the population, the genetic exchange between these populations, and that has some pretty drastic consequences for the health of the gene pools for both of these populations, and it drastically reduces the reproductive success of birds in these habitats, and we'll talk more about that later. All right, now let's move on to the last topic of this lecture, genetic drift. Genetic drift, the name here is actually a very good description of what's going on. But genetic drift, I found, is one of the more difficult concepts for students to understand. Of the four evolutionary mechanisms, this is the one that they struggle with the most. Another term used for genetic drift is random drift, and that's actually even a better description, I think, of what's going on, because, I mean, that's what drift is, right? If you think about something drifting in a lake or a pond, it's not moving in a predictable direction. You can't predict in the next time frame where a leaf is going to be if it's drifting. So it's random movement in that example. From a genetic point of view, we're talking about random changes in allele frequencies. So generation after generation, the allele frequencies in, say, gene A, you may have two alleles, allele A1 and A2. Does A1 go up in frequency or go down in frequency? You can't predict. There can be random changes in that, and if that's the case, that is most likely due to genetic drift. So this is a non-adaptive evolutionary mechanism, and I want to contrast that to natural selection. Remember, natural selection was a non-random process. You can predict what is going to happen with allele frequencies if you know what the selection pressure is. But you can't predict how which alleles are going to increase in frequency or decrease in frequency with genetic drift. There's only one thing that you can predict with genetic drift, and that is the magnitude of its effect. Genetic drift or random drift is much more likely to cause major changes in small populations. So small populations or small samples of populations, when a population goes small, maybe from large to small, you're going to have some major changes in allele frequencies. You can predict that. What those changes are going to be, you can't predict, but you can just talk about the magnitude of the change is going to be inverse to the size of the population. So, really, this is based on the mathematical principle of sampling error. So let's just talk about that at first to maybe have you have a better intuitive sense of what's going on here. And the best example of this is tossing a coin. Tossing a coin. So you should know, right? If you flip a quarter or half dollar, you should have equal probabilities of getting heads or tails at each toss. So just because I flip heads this time doesn't mean I'm going to get tails next time. Every flip is independent of the other, and the probability is not going to change. So the outcome of one toss is independent of the previous toss. But you also can expect that if you flip the coin lots of times, where you'll get a representative sample, you should get about 50% heads and 50% tails. And that's the key. The larger the number of flips, the more likely you are to see a 50-50 ratio. The smaller your sample, the fewer flips you have, the more likely you're to get some deviations from the 50-50 expectation. So, let's say you flip a coin 5 times versus 100 times. If you flip a coin 5 times, if someone comes up to you and says, I'm going to flip this coin 5 times and get heads every time. I'll bet you $20. That's, the odds are in your favor that one of those times is not going to be heads. Okay, but would it be something to write home about if someone did this? No. I mean, if you flip a coin enough times, you're going to get 5 in a row, right? Now, the same person comes up to you, so you might be able to do that, but somebody comes up to you and says, I'm going to flip this coin 100 times to get heads every time, bet you $20. You take that bet, right? I mean, because the probability of that is, it's not impossible, but it is so much more unlikely. And the reason for that is you have a bigger sample size. You're more likely to get the expected 50-50 ratio or closer to it with a greater sample size. Small sample size, weird things can happen. And this is basically the same thing that's happening at the genetic level with evolution. So let's say that we start off with a population that the initial allele frequency over here, where we've got all these sperm and the eggs that are represented either by A1s or A2s, and that the initial allele frequency in this population is 60% of these are A1, 40% of them are A2. All right? And let's say that you just randomly, let's say that you have put this up like a dartboard, and you threw 20 darts to select 20 gametes that are going to form 10 zygotes, and see what the next generation is going to be based upon your random selection of these gametes. All right, well, that's a small sample size, so we are showing drift going on here. And in the coming weeks, I'm going to be showing lectures that are going to show a very similar kind of scheme associated with graphics like this. So when you see some symbol like this means that drift is playing a role here. So we say that the number of zygotes perhaps that you sample that formed A1, 6 individuals of the 10 zygotes formed. 2 of them were heterozygous, A1, A2, and 2 of them had A2, A2. Okay, this is just, again, because your darts just randomly pick these. All right, well, let's then look to see how many of these zygotes likely would have grown up to be juveniles. It's the same ratio. Okay, so that what we're indicating here is there was no selection. One phenotype or genotype of these zygotes isn't more likely to survive to become a juvenile, and the same thing for growing up to being adults. No difference there. So at the end here, when you look at the frequency of the A1 and the A2 alleles, well, now we have 70% A1, 30% A2. That's a pretty big difference between these. And why is that? Was it because the A1 was a better allele? No. Again, there was no selection here occurring where that if you have an A1 allele for somehow you have a selective advantage. There's no way that it is an adaptation in this example. So what explains this evolution? Because we do see evolution here. We have, remember, evolution is the change in allele frequencies from generation to generation. We've got a change here, a pretty substantial change. The reason for this can easily be explained simply because you didn't pick very many. You just picked 20 gametes to form 10 individuals. That's a small number, and it's not surprising that you're going to get some deviations from expectations of it being the same, but not evolving. And if you did this a whole bunch of times, if you just played darts all day and you kept track of the frequency of the alleles, you'd come up with a distribution like this. So about 18% of the time, so this was done with a computer sampling this, hundreds of times, and you come up with this distribution. The probability that the new frequency is going to stay the same, so A1 is going to be 0.6 and A2 would be 0.4, 18% of the time that would be the case. But the majority of the time, you're either going to get a decrease in A1 or an increase in A1. Okay, and that is if you stick with that sampling protocol of just picking 10 zygotes. 20 gametes to make 10 zygotes. But let's say that you got a little ambitious and you started throwing darts, and each time you were going to pick more gametes to form a greater number of zygotes. If you did that a number of times, this is what you would find. The greater the number of zygotes made, the closer you are to staying at that 60%. Here's one test of that, computer simulation of it. Here's another one. So what you notice is at these small population sizes, where you're not sampling a lot, in this case, boy, it really jumped up where A1 increased, jumped up A1 increasing here. Boy, at least initially, A1 really dropped. It then increased, but then it dropped again. So this is just totally random, right? Within these simulations, you see random changes at low numbers. And then across the simulations, you can't show a similarity among these. The only thing you can say that is the same among these simulations is the greater the number of zygotes you pick, the more likely it's to be right on that 60% line. Okay, the smaller the number of zygotes picked, the smaller the sample size, the greater the deviation from your initial starting point of 0.6. So let's talk about this, how it could affect populations in nature. We're getting close to hurricane season here on the Gulf Coast. And let's say that a hurricane comes through Galveston and you have a population of birds and only 100 out of 1,000 individuals survive a hurricane. Well, this is going to randomly eliminate some alleles due to the death of a substantial number of these individuals and very few survivors. If, however, only 500 die, so instead of a 90% reduction in the population, you have a 50% reduction in the population, there's more likely going to be a greater representation of the original allelic diversity. So you're going to have less of an effect of genetic drift in this circumstance. So why am I using a hurricane as an example of this? Because if you're a bird and you survive a hurricane or not, probably doesn't have anything to do with natural selection, right? 2x4 is flying through the air, debris flying through the air, and if it whacks you in the head or not, that's just a total random event. There are not anti-2x4 or 2x4 ducking alleles that are likely to explain who survives and who doesn't. Who survives and who doesn't in this example is most likely be due simply to luck. Okay, so that is randomness. All right, so genetic drift, remember, has a much stronger effect on small populations. So let's just give a made-up example of this. Let's say we have a population of birds with 2 alleles for maybe some patch on the wing, brown and green. Let's say we start off with two populations, one large population, one small population, and that say 50% of them die in both cases. Well, what's going to be the effect of this? Well, for the large population, really nothing, right? I mean, we have a substantial number of individuals dying because it was a large population, 50% of them die. But you started off with a lot of individuals, and so there's still likely to be the preservation of both of these alleles if they were evenly represented among these individuals in the original population. Now over here in the small population, we still had even representation of both of these alleles, but we didn't have very many individuals. And so if half of those die, there's a chance one of these genes is just going to go extinct. Okay. Again, does it matter whether you have a brown patch on your wing or a green patch on your wing, whether you survive some tragedy? No, probably not, right? So this is just totally random. If you do this many times, you may not always see one allele go extinct. Sometimes you may see the green allele go extinct. Sometimes the brown allele go extinct. Sometimes they'll both be represented. Probably not going to be represented in 50-50 fashion in most cases, though. Because you're dealing with a small population, you're likely to get some changes. That's the point. Much more likely to be the case than if you're dealing with starting off with a large population. So there are a couple of specific terms associated with patterns of genetic drift. One is which is called the founder effect. Let's say that you have a new population founded by a small number of individuals. So let's say that this is again, a population of birds that have the green allele and the brown allele. It's a big continental population. A few of these individuals get blown off in a storm and found this new colony on an island nearby, or they've for whatever reason, they end up on this island. It's a rare event. You don't see normal movement of birds between this island, so it's kind of a one-time event. Well, there's a decent chance that this new population is not a perfect representation of the genetic diversity associated with the original mainland population. Because if you only have 4 individuals making out here, well, all of those individuals may have just had been homozygous for the brown allele. So because the founding population is smaller, it has a smaller genetic pool, and it tends to be much lower in genetic diversity compared to the original much larger population. So we call that the founder effect. Here's a numeric example of this. Let's say that the original continental population had 15 alleles represented at this one gene locus. So very highly polymorphic, kind of an extreme example, but let's just say that's the case. There were 15 alleles. Now obviously each individual only has 2 alleles, right, in a diploid organism. But across the entire population, because it's so big, there are 15 alleles that are possible in the entire gene pool. Well, let's say that 5 birds established this new population. At maximum, the new population can only have 10 alleles. And that's assuming that each of the birds that made this new population, that founded this new population, were heterozygous, and they were heterozygous for different alleles. So that's the best case scenario for maintaining genetic diversity, and it's already dropped by 1/3. It's much more likely that some of the birds were going to have some of the similar alleles, and it's going to drop even more. So you're going to have much lower genetic diversity on the island compared to the mainland population. That's how founder effect works. So in this just random example, these alleles did make it to that population, but it probably is even going to be more extreme. So here's a real example of this. Silver eyes are a population of birds that are found in Australia, Southeast Asia, different species of silver eyes and white eyes. And they have established new populations on different islands from the mainland of Australia. And as they have landed on each island and established populations, starting off in Tasmania, then moving to New Zealand, different islands in New Zealand. Every time they do so, if you look at the original mainland population's allelic diversity, the number of alleles that they have, and so kind of is close to my previous example there. So they're maybe around 12 on average for the population, sometimes higher and lower. As you go to Tasmania, look at the drop. Okay, and then as you go to more and more subsampling, you get lower and lower genetic diversity. So every time you establish a new population, you lose some allelic diversity. So founder effect is one way that we can see the operation of genetic drift. But if populations for some reason shrink rapidly, you also see genetic variation drop. So again, think about a natural disaster like a hurricane that may kill off the majority of individuals in a population. Now, another interesting fact that can be associated with genetic drift is that what was formerly a rare allele may just be the lucky allele that survives one of these drastic reduction events, which sometimes we would refer to as a bottleneck event, or from a founder event situation. And this rare allele now becomes common in the surviving population, or the new population survives on the island. So again, let's say that we have this population that's almost 50-50 between the brown allele and the green allele, but you get this mutant that becomes established in the population that's less than 1%. But that one individual may be what survives the hurricane, or is represented in one of the individuals that founds the new population. And in a very short period of time, it can go from less than 1%, extremely rare, to maybe 25%. So much more common. And again, this is one possibility. I'm not saying that this is a common thing, but it's a possibility. And it's not, again, I have to emphasize, this is not an example of natural selection. This rare allele is not now somehow the most beneficial. So it's the reason that these individuals founded this population, or the reason these individuals survived this natural disaster. If that was the case, then it wasn't due to genetic drift. What I'm saying is, if a small population does survive the disaster or found the population, if it's just random who does that, a rare allele can increase in frequency. So give an example of that. So on this island northeast of New Guinea, Pingalap Atoll, the people that lived on this island in 1775, the population was decimated by a typhoon. And typhoon basically the same thing as a hurricane, different part of the world, they call them different things. Well, 20 survivors of this storm, and one of them was heterozygous for achromatopsia. So what this is it's associated with complete color blindness. It also leads to light sensitivity and poor visual acuity. So we're not talking about something that is selectively advantageous. This is not an adaptation if you're living on this island. But this rare allele suddenly became common. So it was probably very rare in the population, but then 1 out of 20 individuals that survived had it, so it instantly became more common. And even in today's population, 1 in 20 has this achromatopsia. And in most populations, it's much rarer that maybe 1 in 20,000 will have this. So that's an example of how genetic drift can lead to a rare allele increasing in frequency, and importantly, and this is going to be very important when we talk about conservation genetics, even if this allele is not only not adaptive, but it actually is damaging to fitness in some way. Okay, so the next video is going to focus on how these 4 evolutionary mechanisms can influence genetic change from generation to generation, and how we as scientists can quantify that. So if you get a certain percentage of allele frequency change from one generation to the next generation, how do you quantify that change and say, yeah, there's been evolution that's occurred here, or no, that small change really isn't significant? And so we'll introduce the concept of what's called Hardy-Weinberg equilibrium.